Hey, what is going on? It is Friday, March 11th, 2022, and I am excited to finally move forward within this glycolysis process. I feel like I've been going back and forth between step 1, step 2, and step 3 for quite a bit, and it's only because I've been really solidifying my knowledge about how glycolysis works and trying to get familiar with all of the inhibition and activation actors in glycolysis and the various enzymes that are involved, because inhibition and activation are very important. Regulation of glycolysis is very important to the cell because as glycolysis goes forward, so do the energy outputs as well. And if the cell doesn't need energy or does need energy, that is that's glycolysis is purpose in a nutshell. So anyway, enough about that. So I'm going to talk about step 4 of glycolysis today a bit, um, and uh, and then just kind of introduce the actors and their the products and the reactants, and then go into the details in another video. So just kind of to recall where we are with glycolysis right now, and I think in general it makes sense to do this recollection as we go through these videos. Uh, Remember, what happens in the beginning is that glucose comes through the GLUT3, the GLUT transporters, right? GLUT1, GLUT2, depending on where the glucose is, what type of cells the glucose is being pulled into. And that's done through facilitated diffusion across a concentration gradient. Um, Glucose comes through those transporters and is immediately phosphorylated by an enzyme called hexokinase. Remember, kinases do phosphorylation. Okay, so I like this diagram because it's showing exactly what's going on. So a phosphate group is added to glucose, and we get glucose 6 phosphate. Right. So once we have glucose 6 phosphate is a polarized version of glucose that cannot leave the cell because the GLUT transporter is only reacts and changes its conformation to glucose, not a which is and not a polar version of glucose. Also, glucose 6 phosphate can't diffuse outside of the phospholipid bilayer of the internal cell membrane either, so it's stuck in the cell at this point, right? So at that point, the next thing that we talked about was the isomerization of glucose via phosphoglucose isomerase. Okay, so that's basically the conversion of glucose 6-phosphate to fructose 6-phosphate. Now, yeah, I mean, you might be wondering why logically this would happen, right? And we'll talk a little bit about that. But this is just a review. Fructose 6 phosphate is then by the action on step 3 by the action of fructose kinase, phosphofructose kinase PFK1 and PFK2 um, are is converted to fructose 1,6 bisphosphate. So it's a doubly phosphorylated fructose. Okay, so also to note that this is just the carbon structure in terms of the straight chain. At this point, the fructose is a sugar, is a ring sugar, same as the glucose. So this is just kind of the chemical composition, a rough chemical composition, and it's uh, don't get fooled, it's not a straight chain molecule at this point. It's only temporarily a straight chain molecule once it's opened up uh, by the enzyme, so it can be converted to fructose. So this is basically the third step, and that's what we've gone over so far. These steps, again, are regulated by the presence of allosteric inhibitors, which include fructose 2,6 bisphosphate, also ATP and ADP, will regulate the activity of PFK1 and PFK2. And even there's hormonal inhibition slash activation via insulin and glucagon. And hormonal activation and inhibition, just a slight tangent, those are occurring because there are processes outside of glycolysis within the cell, which within the body, right? Because remember where insulin and glucagon come from, the pancreas, essentially. Um, the those processes outside of the cell uh, have a vested interest in how much glucose is converted to fructose, aside from the cell's immediate energy needs. So the evolution has sort of created this hormonal interaction with these enzymes so that the enzymes can also stop or continue or increase or accelerate or decelerate their activity based on the presence of these hormones like insulin, etc. So that's just a quick note. And then now I'm just going to mention quickly, without going too deep into it, where we are. And so we are at step 4, where the enzyme known as aldolase, right, will basically break down aldo, right, will break down fructose 6, 1,6 bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, or DHAP and GA3P. So you've probably seen DHAP in the store and GA3P in the store as in the form of a supplement. It's probably because they were involved in this process, and whether you take them raw or whatnot, I don't know. I'm not a doctor. I have no idea. But I wouldn't think it's as simple as taking raw versions of these and saying that, oh, okay, now the rate of glycolysis production is now affected via Le Chatelier's principle. I wouldn't think it's that simple. But that's just something, that's always been in my mind. But let's see why this happens, right? We really, the end of the day, we want 2 pyruvate molecules, right? And essentially, by just as a reminder, what happens here, right, is that the molecules are split up. The 6 carbons split up into two different 3-carbon molecules. The difference is basically one molecule is a ketone, dihydroxyacetone phosphate, ketone phosphate, and the other is an aldehyde phosphate. So it's the ketone is dependent on where the oxygen here, the carbonyl oxygen is in the center of these 2 groups here, and the aldehyde phosphate has the carbonyl oxygen at the end of the glyceraldehyde 3 phosphate molecule. So yeah, that's just a note. Now, skipping ahead a tiny bit, in order to we essentially are looking to get 2 pyruvates, right? Dihydroxyacetone phosphate actually has to get converted via an enzymatic reaction to G3P. Okay, that's why this is even present. So there is another step in which that happens, an intermediate enzyme which takes care of that, and the conversion of that has implications for the fate of pyruvate down the line. And we'll talk about that later. But whether pyruvate essentially in the presence of oxygen becomes lactic acid, or whether it becomes an alcohol, whether the end product is alcohol via fermentation, those are going to be important. But for now, we'll just need to know that we need 2 pyruvate in order to continue the production of ATP. So that's why this happens. So, but in this step, just to note, aldolase, the aldolase enzyme takes care of this. Aldolase is a complicated enzyme, aldolase. And a lot goes on in it. Specifically, shift-based chemistry occurs in aldolase to do this work via the action of lysine and tyrosine side chains. Okay, so I'm going to go a little more into that later on, but just to kind of talk a little bit about what aldolase is doing, and it's cleaving these 2 molecules, and these 2 molecules then move to the next enzyme. We also want to talk about regulation of this step, and when this aldolase comes into play, what else is needed, and we'll talk about the reaction mechanism later on, because the reaction mechanism is actually quite involved. Okay, so that's it for this short video. I'll see you in the next one. Thank you for watching. Like, subscribe, and yeah, leave a comment if you feel like it. Thanks a lot.